something we got interested in doing during COVID, and we have continued, and I think the people who've attended have found it helpful. But we're delighted to have you here this morning. It's a pleasure for me to introduce Adam Naples, who I've known for many years. Dr. Naples is a real expert in the area of neuromarkers, biological markers of autism. This is a very hot topic area right now. The federal government is looking into new approaches to diagnosis. Sadly, the screeners that we have not ended up working quite as well as we wanted for complicated reasons, which itself is a whole topic. But we're pleased to have Adam this morning, who's going to be talking about biomarkers and autism.
Thank you, Fred. Let me share my screen, and Eileen, you'll tell me if something goes wrong. I hope. I don't hope anything goes wrong. I hope that nothing goes wrong, but you'll tell me if it is. All right, thanks. Thank you for the introduction, Dr. Volkmar. I mean, I think you really gave the best summary I could give of this talk, which is that, many of the screeners and tools that we have, many of the tools and screeners we have are really effective in some ways and not as effective as we would hope they would be in some other ways. And I'm gonna give you a little story about that. A little bit about my background. I'm a cognitive psychologist by training. My research is focused on using measurement and methods from cognitive psychology, cognitive neuroscience to understand individual differences. I'm going to talk mostly about our work in studying and identifying and evaluating biomarkers in autism. And I'm going to shoot for about 40 minutes and leave some time for questions. All right, so we're going to talk about what biomarkers are, why they're important, and some studies of biomarker research in autism. And then I'm going to give you a little bit of a teaser about some more inclusive experimental approaches that we're developing for biomarker research and some novel biomarkers that we're developing to study social cognition. So, what's a biomarker? So a biomarker broadly, it's a defined characteristic that's measured as an indicator of a process, a normal process, a pathogenic process, a response to an exposure and intervention, and that includes therapeutic interventions. And so when we think about biomarkers, I guess heart rate, like blood glucose, blood pressure, these are measures. These are things, measures. I don't know that many of us think about them as biomarkers for that word if you don't do this kind of research, but they're routine measurements of a process. They're reliable and they're objective. And if anyone wants to read a lot of compelling writing about biomarkers, the biomarker working group right up from the FDA at the bottom there is actually pretty great. But why do we need biomarkers? Well, most of the time we want them, and I'm not just talking about psychiatric biomarkers here, we want them to make decisions, right? Like we want them to help us decide, should we, in the case of like maybe reading disability, should I enroll my kid in an early phonological awareness intervention or maybe a reading fluency intervention? We want them for diagnoses before, early diagnoses, early intervention. We want them to help us select treatments. We want them to help us pick the right treatment for the right person. And then we want to know if the treatment that we have chosen is working. And these are all difficult problems, and they're all difficult not just in autism, but we want to be able to make the right decisions, and we want the data that helps us do that. I guess a really important part about this is that we want a good measure for one person at a time, and that's a lot harder than it sounds, because most research that we do it across hundreds and thousands of people. But when someone goes into their doctor, they go see Dr. Volkmar, it's one person, and they have their individual challenges in whatever that domain that might be. But let's talk about heart rate as an example for a biomarker, right? It's good, it's routine, right? Like we can monitor our heart rate continuously on our watches. We can use them for treatment selections, depending on what our heart rate is telling us. And at a given time. We can use it to identify individualized treatments, right? So my old watch would monitor my heart rate continuously, and it would tell me what kind of run I should go for and how long I should run for. And it can tell me if it was working. And so as you can see here, it was, suggesting that my training wasn't as productive as it could have been. And so I give this as an example of what would be really great for all of our biomarkers. At this individual level, we're able to monitor something reliably, continuously, routinely, objectively. And we can use this information to tell us, to guide us on, how we should go for a run in this case. And then we can use these same markers to see how it's going, right? To help us make decisions. Maybe we should rest today. Maybe we should go for a longer run. But now we're going to talk about biomarkers for autism. And also I want to highlight that why we might want to look at biomarkers that are specifically for social information processing. All right, so this is very familiar to many of you guys. These are the DSM criteria for autism. I want to highlight a few things here. Is that one, persistent challenges in social communications have been sort of central to the diagnostic criteria from the start. But they're not the only criteria. So we have what are called restrictive repetitive patterns of behavior, and those often include sensory sensitivities too. But what's important for much biomarker research is this reality that there's a very heterogeneous presentation. And secondly, that our current tools for assessment and measurement are subjective report. And these are really, really, really very good subjective reports there. They include screeners that have been validated in very large samples, very diagnoses by expert clinicians, but they're still the subjective measures. So when we're thinking about biomarkers, and I gave you examples of heart rate, blood glucose before, we oftentimes think of, why is blood glucose important? Why is heart rate important? So when we think about biomarkers for autism, we're thinking about what are these theories behind our candidate biomarkers? What are the theories for what leads to these challenges that people with autism have? And can we use those as sort of clues to help us, decide on what are some good biomarkers? Looking at my time here. Eileen, do tell me if I'm talking too fast or too slow. But broadly, I'm going to go through these kind of quick, and these are our very large, almost meta theories of differences in autism. And it's more important that you appreciate the breadth of these theories and how they reflect sort of different levels of abstraction than really, really, really specific details about any of them. But at the end, I'm happy to answer questions about those specific details. So, right, social motivation, right? There's this theory in that's sort of many different variants of it, that some autistic people are less rewarded or motivated to interact with other people, and that this is something that occurs early on in development, right? And so this reduced motivation to interact with other people, that social interactions are less rewarding, leads to a developmental trajectory where social interactions are not something that people become expert in navigating, right? And leading to maybe reduced social reward, reduced social desire, and importantly, reduced learning about social interactions. So now, another theory that's important as we think about learning is this idea of predictive coding. And predictive coding is a particularly jargony term, but just sort of hold on to it for a minute. It's all about how the brain processes all kinds of information and not just social information. And what we understand about the brain and learning is that our brains are constantly generating implicit predictions about the environment, right? These are not explicit predictions that we're thinking, well, this is going to happen next, or that's going to happen next. But they're things like, if I drop something, it's going to fall, right? If I held this up and I dropped it and it didn't fall, then I would be surprised. But these kinds of predictions, right, these expectations that our mind and our brain is continuously generating, they guide how we learn about things, right? They guide how we look, or even how we look around at different things from second to second, or from millisecond to millisecond. And there's some evidence that people with autism, generate these predictions in a different way. And this leads to different patterns of attention about the world and different ways of learning about the world. Now, this is also a way we could think about how social symptoms emerge, but also sensory symptoms emerge. Because we're not just limiting the way we're not limiting our conceptualization about learning to a specific domain, such as social or sensory. It's just everything that goes in through our ears and our eyes is what guides our learning. Okay, so now for another sort of big meta theory is associated with dysregulated arousal. Now, when we think about arousal, we may have different ideas about what we mean with this word, but here I'm talking about an overall level of brain and body sort of state, right? And these range in extremes from sort of panic and sweating and elevated heart rate to sleep, essentially. And this theory about dysregulated arousal has been borne out in evidence in a variety of domains, and particularly some things that have been more difficult to explain with other theories, like that some people with autism seem to have less sensitivity to things like pain, which almost everyone, has a lot of sensitivity to, and increased sensitivity to other kinds of stimuli like sound, but also things like dysregulated sleep and heart rate. So this is it's even broader, I guess, than predictive coding in the sense that it's really kind of encompassing full sort of brain body state of affairs. And I put this reference at the bottom because there's evidence that people have been seeing evidence for dysregulated different arousal for decades now. And these are some great papers from 1961, 64 they're classics. They're nice and short too. And then finally, there's again, in another theory, there's evidence for a dysregulation or an imbalance of excitation and inhibition in the brain. And this is now where we can get even more technical than predictive coding. But excitation and inhibition is how neurons communicate information in the brain. Some neurons excite other neurons, some neurons inhibit other neurons. And the brain is constantly in a state of flux, or rather a state of sort of a dynamic balance of excitation and inhibition. And this impacts every computation in the brain. I say symptom domains here, but this balance of excitation and inhibition impacts everything your brain does. But it's particularly associated with differences in sensory sensitivity, right? Look, so is the sound heard or is the sound not heard even? Motor performance, cognition. In the extreme, too much excitation is associated with the occurrence of seizures. Too much inhibition is associated with people falling asleep and being insensate to sensory input. That's a lot of theories, and now here's where I hope I can bring them together. They're not all mutually exclusive, and if you've read the literature now for decades, you'll see like a lot of these things kind of seem similar. It's not like social motivation is a different thing than the excitation and inhibition balance. And so, like I said, I'm a cognitive psychologist. I like to think about information processing systems. So here's a meta theory to help guide how we think about these theories. In 1982, this is a great book. This great book came out about vision, and it gave us a useful rubric for thinking about cognition that I'm going to use here. And that's that information processing can be thought of as three levels, right? The computational problem, which is what's the system trying to solve, right? Is the system trying to solve, and the system being the brain, is it trying to solve how to navigate a social interaction, right? Like, what's the problem? I need to get on the bus. I need to go on a date. I need to, tell my kid to do his homework. And then there's the algorithmic level, right? And this algorithmic level, those are the steps the system uses to solve the problem. Like and we think about it just like, all right, first, get the phone and call your kid and tell him to come home and do his homework. 2, when he gets home, do this. This is the algorithmic level, right? Now, lots of different algorithms could be used to solve the same computational problem. And then finally, there's the implementational level, and that's the hardware, which in our case is a brain. That's the actual thing, the physical stuff that needs to implement the algorithm and the computation. This is not the only way or the best way to think about cognition, but I think it's helpful to think about how these theories are going to help us think about the biomarkers that, manage this level of abstraction. So if we embed these theories into this meta theory, we find that they hit at these different levels of abstraction, and they're not at odds with each other. We can think about social brain and the computational approach is learning about other people, making friends. These are the kinds of problems that, when I say problem, I mean sort of maybe goals is a better way to put it. The algorithmic approach, right? Like, okay, well, if we're learning about other people, we have to make an eye movement so that we actually look at the other person and see them and get information about them. And then when we think about the implementational level, right, now we're getting down into issues of arousal and excitation and inhibition. To make that eye movement, we need to have different patterns of cortical cells firing, different sort of brain and body states to guide how information is coming into the system and being processed. These theories generate a lot of hypotheses, and these hypotheses about how people will act. And so now we'll sort of see how they worked. And now thus we get to biomarker measurement in autism. And then before I go right into the kinds of biomarkers I'm going to talk about, it's important to talk about the tools that we use to measure the biomarkers, because they're critical for really understanding when we talk about measurement, like what these things are, what we're measuring. And so there's two tools that are important. One is eye tracking and one is EEG. And what's great about them for biomarkers is that they are inexpensive, right? They're relatively inexpensive. An EEG system, I don't personally own an EEG system, but every hospital has lots and lots of EEG amplifiers. They're easy to use. You don't need to be an expert with decades and decades of training to use an EEG system. I could teach you how to do this in a few hours. They're full of information, and they're objective, right? So they represent our most promising, we think, maybe not the most promising, but very, very promising methodological tools for understanding brain and behavior. Let's talk about EEG. What is EEG? Well, on the left, I'm going to show you some beautiful pictures of neurons that are, I think I have a poster of that behind me. EEG is the sum of the ongoing cortical activity recorded at the scalp that reflects this balance of excitatory and inhibitory postsynaptic activity at the cortex. And we measure this, and this is on the right, my son, but he's not this little anymore. But we measure it with this soft, spongy cap that we put on your head. And each one of those little circles that you can see that's sort of like poking in there has an electrode, and we amplify those very, very, very faint electrical signals generated by your brain into a recording. And this is what that recording looks like. Each one of these wiggly lines from the left to the right is the voltage measured at your scalp. Can you see my mouse, Eileen? If you let me know if you can't, so that I'll stop trying to draw with it. Each one of these wiggly lines is the voltage measured at your scalp from an electrode. Lots of lines means we're using lots and lots of electrodes. And we can learn a lot of things from the ongoing EEG, even when you're sitting around doing nothing, right? One way we can look at this data, which is particularly exciting and is objective and inexpensive and routine, is look at this balance of excitation and inhibition. Right? And I still think this is cool, but I've given this slide lots of times. Those different excitatory and inhibitory neurotransmitters, they spend different amounts of time at each cell, telling it to not inhibit or telling it to inhibit or not inhibit. And that different amount of time means that those wiggly lines are of different speeds for excitation and inhibition, that's so great. We can just measure that from like a little like wire in your scalp. So having people sit and do nothing is actually it's a great way to measure the balance of excitation and inhibition non-invasively. So what I'm going to show you here is called the power spectral density. It's a plot of how big the wiggles are at different speeds. So on the left are the slow wiggles, and on the right are the fast ones. And in general, this is from lots and lots of people. So it's depending on your screen, it might be a little hard to see. There's two kind of thicker lines in the middle, and then those represent different diagnostic groups. And this sort of blur around the edges is everybody's individual wiggle. So you'll see that they're all kind of the same pattern. Everybody has mostly more high amplitude, lower frequency activity. That's the slow big wiggles on the left, and less high frequency excitatory activity. That's the faster wiggles is what we're measuring on the y-axis on the right. So what this means is that this slope of the EEG, the balance of the low frequency activity and the high frequency activity, can tell us what the balance of excitation and inhibition is in an individual person. And shallower slopes are more excitation for a person, and steeper slopes are more inhibition for a person. And this is it's a great biomarker, right? We can put it on somebody's head when they're just sitting there watching a screensaver on a computer screen, and we can measure this in about 2 minutes. And it really does reflect many things we like about biomarkers. Like if we give someone a drug or like Ambien that changes the balance of inhibition to excitation, this whole slope changes, right? So it really reliably reflects this biological process. So we'll get back to the slope of the EEG in a minute. The other thing we can do from the EEG, and some of you might be really familiar with this slide, and some of you probably have never seen it before, is we can derive something called an event-related potential. And so the event is something we show people, and whether that can be, in this case, pictures of faces and pictures of houses on a computer screen. And we can take this on, so this wiggle right here, and again, I hope people can see these things, and this is where we might have to come back later just for some pictures to zoom in. This is like the ongoing EEG. Remember, it's lots of just this sort of wiggle. It's the voltage being generated from your brain across time. Now, what we can do though is we can show you, or in this case, pictures, but they could be sounds or something else. Pictures of faces over and over again, right? We call these events, and then we chop up the pieces of the EEG, right? Like we look at only those parts of the EEG around the event, and apart they kind of don't look like anything. They just look like little wiggles. But if we average them all together, like every time we saw a face and every time we saw a house, we attenuate the parts of that signal that are irrelevant, and we accentuate the relevant parts. And then you end up with this kind of, on the right, what's called an event-related potential. And it has relatively few like peaks, and we call the negative parts peaks too. And these peaks themselves reflect features that can inform us about cognitive processing. And this one right here is particularly important. So some of them are very important for social cognition, and we call this the N170. So here's time on the bottom, and these are time scales that most of us aren't used to thinking about. But this is 500 milliseconds here. So this is about as long as it takes for you to move your eyes, right? These are very, 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 very fast, fast occurring things. And so if we show you faces, more than any other kind of visual thing we can show people, at around 170 milliseconds, there's this negative peak. And this black line is for faces, this gray line is for not faces, in this case, houses. It's really fast, and it's earlier for faces than anything else. And now we think, why is it important? Well, social interactions are really, really, really fast, right? Like if you meet someone and you smile at them and you hope they smile back, that happens in less than 500 milliseconds. So if we have some differences in how these processes are happening at the individual level, we might be able to capture those differences in these most earliest stages of face perception. So this early peak, this N170, it's very sensitive to faces. It's temporally very early. It's very sensitive to context, right? Like if you know the person that you're looking at. And it's sensitive to gaze. It changes too if someone's looking at you or looking away from you. So what do we know about these EEG measures in autism thus far? Well, there's evidence for delayed N170s, that these N170s actually occur slightly later in autistic people than people without autism. And also that the N170 might be of less amplitude. Now, it's important, as I mentioned before, because this occurs so early on, temporally. And then atypical spectral profile, I'm referring to the slope of the power spectrum, that EEG slope, the balance of excitation and inhibition. And it's a very heterogeneous pattern of findings actually in autism. But they're regularly, across studies, there's evidence that the slope is either too shallow, meaning that there's more inhibition or, more excitation, or too steep, indicating more inhibition. The N170, I would say across almost every biomarker studied in autism is probably the most consistent. But with that said, there's a lot of variability in the literature. Now we're going to talk about eye tracking. So eye tracking at a very high level is where we take a video of a person's eye and we use it to figure out with a very high spatial and temporal precision where they're looking. And I'll say that maybe 22 years ago, I saw an amazing talk by Dr. Volkmar about eye tracking and autism, and I hope that I do it justice when I show some of the data from that here. But here's what our eye tracking data look like. And again, it's important, I think, for everyone to have an understanding of what actually the measures are that we get that at the level of the numbers and the data to understand how these things impact our understanding of biomarkers. So all eye tracking data is we have a spreadsheet really with time and where people looked on a screen, and these are X and Y coordinates. And we can find that we can learn a tremendous amount from this, right? We can learn where someone's looking when we show them a social scene, for example. And this dot sort of bouncing around is a marker of where someone's looking at these pictures of kids sledding. At the bottom left, you'll see a video of an eye bouncing around. And the bottom right, this is over time, the X and the Y coordinates of where the eye is looking, right? So it's very, very simple data from one perspective, but it's tremendously rich. Another thing that we can get from eye tracking, which is there, if you saw the eye down there, is we get the diameter of the pupil. And this is a really sensitive marker of brain state of arousal too. So broadly, eye tracking findings in ASD, and this one slide does not do justice to the breadth of the research, but there's very consistent evidence and indicating that people with autism look less at social information, and that's across a range of stimuli in participants. So that's been shown in static images of faces, as you can see in the top right, and videos of movies that are socially rich. That's just a screenshot from the video in the bottom right. And there's also evidence for attenuated pupil constriction to light, which suggests that there is increased arousal as people will sort of want to stay open more with increased arousal. So when you flash a light, it doesn't constrict as much. All right, so briefly, these putative biomarkers in the past have targeted different levels of abstraction and theories, social motivation, EI imbalance. The results primarily reflect modest sample sizes. We have lots of inconsistent findings, but before we can use them as a measure, right, we really need to see which ones hold up. I'm going to speed up now because I've been going now too slow. So I'm going to talk to you about the Biomarkers Consortium for Clinical Trials. So the Biomarkers Consortium for Clinical Trials, the idea here is that there's lots of evidence in small samples for biomarkers in eye tracking and EEG, and people have looked at these in small samples for decades now. So we're going to see how they work when we really measure hundreds of people, and we're going to set it up like a clinical trial with a tremendously high amount of methodological rigor. And so the Biomarkers Consortium for Clinical Trials is a study that's run by Jamie McPartland at the Yale Child Study Center. It includes 5 sites: Yale, Boston Children's Hospital, the University of Washington, Children's Hospital of Los Angeles, and Duke University. Currently, it's recruiting for its next phase, but I'm going to talk to you about the first phase, which incorporated 280 kids with autism and 120 kids without autism between the ages of 6 and 11. And so the idea is of the Biomarkers Consortium, though, was to look at some of those promising markers I brought up and now really, really test them in a rigorous way across a lot of people. So what did our experiments look like? Well, we addressed social motivation in 2 ways. First, we just showed people pictures of faces, like that ERP experiment I showed you before. And this is what our experiment looks like, right? In a way, this is the biomarker this is part and parcel of it. This is how we measure it. We show you faces to elicit that signal, and we measure that signal. We also, what's called, we call it it has a very complicated name, the composite ocular motor index, which is a fairly complicated way of saying, like, how much do people look at faces in socially rich scenes? So these are some of the videos and static images we show people. And the colors around them reflect kind of the regions we measure. Like, we measure how much people are looking at the faces of the scene, how much they're looking at the bodies of the scene. We also measure the pupillary light response. And I've talked about that briefly, but the speed and the amplitude of how much your pupil constricts in response to light is a pretty sensitive index of autonomic nervous system function and arousal. And we do that with really simple. You look at a screen, it's got this little thing in the middle that sort of catches your attention. We flash a light. And that's the signal we get. I can come back to that a bit. When we also just measure the excitatory and inhibitory balance through the resting EEG. And this sort of vague picture right here, it's like a little screensaver that we just put on a screen so that people sort of sit at rest without any explicit task, and we just measure their brain activity as they're sitting there. So, all right, so here I'm going to go through the sort of there's a tremendous amount of data. And so when I say abridged results, I could talk for hours and hours and hours about any one of these, and I'm happy to later, but right now we want to be quick. So let's talk about social motivation and N170s. So now, in the largest, most best controlled sample, we're seeing an evidence that people with autism show later N170s. And let me orient you to these pictures a little bit. So on the left, I'm pointing at the N170. This is that waveform I showed you before. On the right, there's this sort of vertical histogram, and you're going to see a few more of these. And what this is showing us is the N170 latency, and then this is the average the N170 latency across the autistic sample, and on the right, the control sample. And this shows you the distributions of the differences across the different groups. And so I believe this is open access if anyone wants to read it, and there's lots and lots and lots and lots of detail about that. But so we're seeing this evidence for less efficient face processing. We're also seeing in evidence for reduced attention to faces, and that's across static and dynamic tasks. So across our movies on the top, these left two columns, and then just static pictures of people, we see these consistent and large effect size differences. Even after controlling for age and cognitive ability. And so this is, again, more evidence for this reduced social motivation, because if you're looking less at people, you learn less about people. But we also see that participants with autism have a slower PLR constriction, right? And this is an index now for broadly different kinds of states of brain. If sympathetic activity is up, if arousal is up, the pupil wants to dilate. And so when we flash a light, if your pupil dilates less, that's a marker that perhaps these measures are that it's a pretty good measure of arousal in the sympathetic nervous system. And finally, in line with the excitatory and inhibitory balance, participants with ASD have shallower EEG slopes and indexing more excitation relative to inhibition compared to the control sample. And so I know that this might be a little bit hard to see, but the slopes of these lines are significantly different. Visualizing that is not quite as dramatic as the eye tracking results. But what's also really interesting about the slope of the EEG is that it's so reliable that we found out that if you measure it in the same person 2 days apart, that the shapes of the overall EEG spectrum are so similar within people that they almost look like fingerprints in a way. And so back to what we're thinking about biomarkers, that means that we measure this one day, it's going to be the same the next day. So it's reliable, and things that are reliable are great measures. I mean, you can imagine if you gave a math test to someone, two different math tests to someone who knows math, you'd want to get the same score from that person. Well, two different, the same kind of math, I guess. And so when we think about biomarker measurement, we want these measures to have the same kind of properties as good educational tests. And so how do we summarize that? How do we put these findings in this sort of context? Well, so we have evidence for reduced social motivation for the Anderson 70 and OMI, increased arousal from the PLR, increased excitation from the EEG, and these biomarkers, it looks like they're indexing these different levels of abstraction, right? These theories aren't mutually exclusive. There's evidence for multiple mechanisms at work. And so now I'm actually going to skip a little bit to get something I think is more important in this well, I think it's all important, but for timeliness. So where are our next steps there? We're replicating a larger sample right now and actively recruiting. We're interested in the long-term stability of these biomarkers for measurement, and we're looking at the feasibility study in a younger age group. And so, but we did find something out, and this is along the way, and this is it's about data loss. And why would we think data loss is important? Well, it's really important because if you can't measure something from someone that you need to measure it from, then it might not be a great biomarker. And I'm going to say these are great. These are great biomarkers, and we're doing some new research to make them great for everybody. But and we'll sort of jump through this. Here's the point. It's that when we do these tests, when we do biomarker measurements, someone's coming in and we're asking them to sit in a boring room that they're unfamiliar with and look at pictures or put this soft spongy cap on their head, but that's probably a little bit different from what they do from day to day. And we find that the kids who maybe had the most challenges, whether it's cognitive ability is measured by IQ or social differences, those were kids who they had more challenges participating in the biomarker studies. We had more data loss. And these are very, very specific technical details about the variable derivation, but the important part, and I'm going to get to that here, and this again might be really hard to see, which is that data loss is increased in autism. And that applies for all of our biomarkers. And this is not unique to our study, but I can quantify it very, very, very well in our study. It is significant in all of the biomarkers and all of the markers of how we look at missing data. And that data quality is associated with the clinical characterization. So again, this giant table, I just want you to look at all these asterisks indicating significant differences. We're losing the most data from the kids who are the most impaired. That's the important part. And that means that those kids who might be in the most need of effective tools and measures are those kids who we are learning the least about in our studies. Now, the data quality is very high, measurement of social tension, most compromised when social tension is reduced. But now I want to talk about biomarkers for inclusion, and this I think is a particularly important feature, which is that it got us thinking about, in the biomarker study, a group of kids who are really in significant need of study, which is kids with autism and intellectual disability. And estimates suggest that approximately 30% of people with autism have significant intellectual disability. They're very, very, very underrepresented in neuroscience research. And this is a slightly outdated slide, but I did this last year. I took from 2020 to 2023, I looked in 3 autism journals that put out a lot of articles. And they published 547 articles since 2020 in that 3-year period. And then we looked across every article we could find in those years, not just in those articles, just every journal we could find in neuroscience journal, to look for studies that included kids with severe intellectual disability in neuroscience and EEG research. And we found that there were 66 kids. That's not 66 studies, that was 66 kids that we read the studies, we looked at the sample sizes, and we added them all up. And I will also say that I suspect that some of these kids were actually participating in multiple studies. So the number is 66. At best, it is 66 kids. That's suggesting that the kids who might need some of the most help are the least included in the research. And why would that be the case? Well, it's hard to get usable data. And when I say usable data, there's all about the measurement things I've been talking about, right? Like if we're going to measure your EEG, we have to put this thing on your head. If we're going to measure eye tracking, you have to sit still in a chair and look at a screen. And so for some of these kids, this is not like people don't know. This is is important, but it's hard, right? We need, specialized staffing. We need, to be able to support the needs of the families and the kids. We need, specialized analytic tools, because the data are typically a little bit noisier, right? Like if we move our heads, we can't measure the EEG when you're moving, for example. But I guess importantly, like it's really hard for the families, right? Their lives are complicated. And so we're asking you to come in, miss a day of school, come to this new place, park in New Haven, which I think for those of you in New Haven know is not a trivial problem. And it's hard for the kids, right? These are kids who have face a lot of challenges, and they come into our lab and we say, sit still and look at a screen quietly for 45 minutes. That's hard, I think, for many people. It's particularly hard if you don't understand what we've said, and that's the case for some of the kids and families. So, I mean, ultimately, it comes down to getting usable data and the task demands of getting usable data, which are typically following verbal instructions, right? Like sit still and look at the screen, sustaining attention, right? So I know that many of you were probably riveted by that faces experiment I showed you before, where faces were popping up on the screen, but not everyone is as engaged and finds that quite that interesting. I also have to tolerate this weird and unfamiliar setting. That's not, it's unfamiliar, and I think it could be weird. We do our very, very best to make these studies as comfortable as possible, but still you're coming to a new place, with a bunch of people you don't know, and we're going to, maybe put this, like, it's a soft spongy cap, but it still has some, saline solution in it. And you have to, understand these spoken or written instructions. Most cognitive neuroscience research is designed and built around college students who most of cognitive neuroscience research is done on, where you show up at a lab, we'll ask you to sit and look at pictures for an hour and a half, and then maybe you'll get $15. Well, you get more now, but when I was in college, that's what would happen. I'd sit and do this boring thing for $15. And it's tough. And when I talk about usable data, these are our wiggles, right, that I showed you before. On the top, that's the EEG measured from someone who's sitting still, right? This is brain activity we're measuring. But if you're moving, right, even if your eyes are moving, this is what you get. It's mostly, you're not measuring brain activity anymore. You're measuring the little bit of like friction from when those electrodes move very, very, very slightly on your scalp. All right, so how do we overcome these hurdles? So we've decided that rather than trying to make things, really hard, is just try to make things easy. So we've made an approach for conducting these experiments that uses computer vision to track movements and real-time processing to sort of help people learn how to sit still and attend to what's going on the screen. It's called Pelican. We thought that was a sort of a cute name, but it adaptively teaches people to attend to the experiment, and it monitors data quality in real time to adapt to trial delivery. There's no explicit instructions, and there's personalized reinforcers. Those last two parts are really, really important. You don't sit down and get told to do something, right? It sort of, unfolds. Here's how it works. So we measure your movement using measures of head movement and attention, where you're looking on the screen. And you come in and we put on sort of a reinforcing video, or just a movie that the kids like to watch. Now, if you move too much, right, and this is if your head is moving too much, if your body's moving too much, and then this movie stops playing. And now it's a big ask to have people sit completely still. So, or to hope that people suddenly figure this out and sit completely still. But if you start moving less, right, and everyone will start moving less because you've been moving a lot, and so you regress to the mean of movement. If you start moving less, then that movie starts playing again, right? And then as you continuously sort of apply this positive reinforcement, the sort of threshold for movement slightly goes down to adapt to this person as they're watching this movie that they've chosen to watch, always positively, so that you're eventually sitting pretty still. And you've learned without verbal instruction, without someone giving you a sheet of paper with what to do, how to sort of that you can sit still and just watch this movie. So we get attention and sort of quiesce or sitting still without explicit verbal instructions. Next, I really hope that Disney doesn't get angry at this, because this is one of the videos a kid wanted to watch. What do we do for unfamiliarity? Before time, we talk to parents, we talk to caregivers, we say, what does your kid like to watch? Like, not the kind of thing, like what do they like specifically? And we'll have that ready. Like, we'll have that queued up in our experiment, so that they come in, that particular thing is what's available. And this has made a huge difference. And I'm going to show you just, what some choices have been. This is, one choice someone had. Another kid loved the intros from 80s TV shows. And so this was very, very, very popular with one family. These are, the specific choices, like, we get ahead of time. And then, someone really enjoyed the Chicago transit bus videos, which we have here. And so I'm going to stop that one so you don't have to hear the video. But the point is that it's so familiar to these families and these kids that it really helps to reduce the unfamiliarity. And then finally, because we're doing all this measurement, right? Because we're measuring where you're looking, when you're sitting still, instead of showing you hundreds and hundreds and hundreds of faces, just hoping that you're sitting still for most of them, we can now determine during the movie how many you were sitting still for. And if you already saw it, the 20 faces, we need to get a good signal. Now we'll show you houses. And the result of this is that the experiment could be even shorter, further reducing burdens on the families. So, and this is just broadly, I want to show you that we've done this successfully with a small sample of experiments that we've adopted from the Biomarkers Consortium to look at social motivations, N170 and these static scenes. And result-wise, it works. I mean, these are trajectories we can measure, when people are moving, when they're seeing the experiments. And it's a very busy figure, but just these little lollipops here are when they're seeing the experiment. The little blue lines are when they're moving, and we can see that some people move a lot, and then they do learn, like, wait, they learn the contingencies, and then some people learn throughout. But the experiments adapt idiosyncratically to the participants. And this is, we've successfully measured these biomarkers at the individual level in these kids, which is I think a tremendous advance, given their underrepresentation in research thus far. And so there's our next steps with that. We're working to deploy this in a larger sample, and some very, very specific eye tracking calibration technical things I'm happy to talk about if anyone has questions. And then this is basically, I'll jump through it, but here's the important part: is many of the interactions social interactions are what we're interested in when we talk about social things. But most of these experiments you've seen is a kid sitting in a room alone, just watching TV, essentially. And so we've developed experiments that actually respond to where you look. So when you look at a face, the face looks back. It can very, very, very basic kind of interaction, but really we think the sort of earliest kind of social interaction you can have, like a baby looks at its mom and the mom looks back. And so it turns out that we can now measure brain activity in these contexts, and we find, and so this is jump through here, in adults with autism, when we look at brain activity to faces that are responsive in predictable and unpredictable ways, that we actually see more, we see large and quite interesting differences when eye contact and social interactions are unpredictable. But we don't really see these differences when eye contact is predictable, suggesting that there's something not just about the content, like faces or eyes, but actually how they unfold in the world. And it's associated with symptoms such as sensory sensitivity. So we'll jump through because I think there's a couple questions there. But basically, in summary, we're finding evidence that these biomarkers are successful at different levels of abstraction and theory. And we're kind of capturing the full silo of implementation from to algorithm to computation. And now we'll jump over that. But if anyone has any questions, I just need to say thank you, because I'm really the hood ornament on a tremendous amount of work here, and acknowledge the families that are the participants in our families the most. And thank you. So, chat question.
Great, thank you very much.
Yeah, I apologize. I mean, I'm happy to. All right, let me answer some of those questions in there to Jackie.
Great.
In the base IQ is 60, were children with profound autism excluded from the study or just not found in the patient participant pool? No, they were not excluded from the study. The base IQ, so that was just to me, were children with profound autism. So someone asked, were children with profound autism excluded from the study or just not found in the patient participant pool? No, they were not excluded. And in that sample I showed you, I showed you the summary statistics. I don't have like right at hand the individual characterization from every participant. But I think one thing that was sobering during that, well, we're still running the study, but one thing I found very sobering is that, these kids are in the ages of 6 to 11 to align with the Biomarkers Consortium. But we also, some older kids came in, 14, 13. Families, some families felt like, well, they were happy to come in, but they were sort of surprised. I think that they had participated in research when their kids were much younger, and they kind of felt like they'd been forgotten as their kids got older, and they weren't. They were like, "Wow, you guys are doing studies our kids can participate in." I guess they weren't just even expecting to be to have these kinds of opportunities, and so I hope that kind of answers the question. We're trying to get the word out. So, and then okay, biomarker research before and after treatment or therapy. I'm not sure what that's particularly what that's referring to. What does it mean for schools and identification implications with current assessment test techniques? I'm not, so I don't work in the schools, so I do know that there's a lot of nuance there that I'm not going to be, that I'm not an expert in. Implications with current assessment techniques. I don't do clinical evaluations and assessments, but I think that everyone wants better tools. And we're trying to provide better tools that people can use. Right now, these biomarkers still are, you're in a lab, you're coming into a lab, you're doing an EEG. But we're trying to understand, basically we're trying to kind of approach from multiple angles how we can make these markers that we have right now into things that are more accessible for practitioners. I hope that was sort of helpful in a vague way. Any other questions? One person has a very specific question across calibration procedures for Pelican. That's someone who I used to work with and who worked on this study, who I miss, and Cassie, you're always welcome to come back and visit. Those are, that would direct, I think there's a lot of math there, and I think it would probably bore people, but I'm happy to talk on Zoom.
We can talk offline later, Adam.
Okay. Thank you. Remote access for the future. Well, actually, yeah, we're about to start a remote access study on auditory perception and auditory sensitivity. That was a direct message to me. And I have some collaborators who are starting an eye tracking study remotely. I think there's a couple of those. We're really excited to get that auditory sensitivity study running, and hopefully within the next month, actually, we'll be bringing people. Well, we won't be bringing people in. I mean, virtually, we'll be bringing people in. There's a lot of yeah, I guess there's a tremendous amount of value in remote access studies. A challenge, particularly in psychophysiological research like EEG or any kind of assessment of attention, hearing, seeing things is really like everybody has a different computer, everybody has different headphones on their phone, and that matters. That can matter a lot in the kinds of measurements that you can get. And so we don't want to find out. Well, I guess maybe we really don't want to introduce bias. And I think that someone in this message, someone mentioned here, historically underrepresented groups. So, I mean, it's a sort of there's a kind of bias I think that a lot of people don't think about, is that different kinds of computers will have different speakers, and more expensive computers will probably have nicer speakers. So if you want to do a study of auditory perception, for example, and you do it remotely, you want to make sure that you don't introduce a bias such that your results reflect the kinds of speakers that people have on their computers and not the differences in how they hear things. And that kind of it's a little bit mundane issue, particularly in like a remote study, it multiplies dramatically. And so it's really, those are the biggest challenges, and that's what we worry about the most in deploying remote studies, is making sure that we don't inadvertently introduce biases like that. But thank you guys for being here. Happy to answer any more questions. My email is there if anyone has any, anything in particular.
Thank you very much. It's a great presentation. We really appreciate it. Thanks, Adam.
Thank you, guys.
Thanks so much, Adam.
I guess we're over. All right. I'll see you later.
Thanks so much, Adam. This was great.
Bye-bye.